Oh, welcome back. Today I'm using a slightly louder speaking tone because my parents aren't home right now and they won't hear me. So I am embarrassment free. I can talk however loud I want. So let's get it started. On the topic of today's agenda, we have my very first tutor video from Tut titled DNA Replication. DNA Replication. Now, seems pretty simple. And it is. Fuck you, textbook, for making it super complex. It isn't. Okay, I'm gonna teach you. So, you got DNA. It's in this twisted double helix thing, twisted ladder. How do you split that up and create two different of the same thing? It just doesn't go out of nowhere. We don't got Hermione from Harry Potter to just go, and then the new DNA pops up. No, we can't do that. We have to do some science. And how do we start that off? Well, how do you start anything off in the body? With an enzyme. And that enzyme is called helicase. What does helicase do? It splits up the helices, okay? It splits up the ladder. It cuts it in half. So now your DNA is into two, not two separate strands, but it creates this crotch in the DNA. It's like a fork. It looks like the letter Y, like this, okay? Helicase creates that fucking crotch and it keeps on going along spreading these apart, all right? So now you have this replication fork. What do you do now? Well, you're going to take an enzyme and that enzyme is going to add the nucleotides to those separate strands. So you can create the two new strands. Wow, it's so simple. Oh wait, it's a little more complex. I'm sorry, it's not my choice. So, that enzyme that adds those nucleotides is called DNA polymerase. There's a like 11 different kinds of DNA polymerases, but we're going to keep it simple. Well, let's say this is DNA polymerase 3. So, DNA polymerase 3 is going to go along adding those nucleotides, but wait. DNA polymerase is lazy as fuck. It doesn't want to start its own job unless someone already started it for it. So, what? Who's gonna start it? Because DNA polymerase doesn't want to start their job. They're too lazy. They can't just go immediately starting adding nucleotides. You know what it needs? It needs a primer. And who's gonna put that primer? Primase. Wonderful naming. Finally, they listen to us. Fuck the photo systems if you know what I'm talking about. Anyway, so primase is going to put a primer in for DNA polymerase to start doing its actual job. And it's great. But there's a catch. Primase, it puts in about 5 to 10 nucleotides. That's the primer. But those nucleotides are RNA. Don't ask me, I don't know why. I couldn't tell you if I wanted to. But those are RNA segments. We're gonna go back to that later with another enzyme called DNA polymerase 1. Well, let's not talk about that right now. So you have the primer, which is 5 to 10 nucleotides that are RNA, and then DNA polymerase can just start off of that and make a bunch of fucking nucleotides and attach it to the string or whatever. So, we have established primase and DNA polymerase. Now that's great and all, but DNA polymerase is a stuck-up bitch, and it only wants to add to the 3' end of the strand. Now, Kevin, you must speak the English. What does this 3' end mean? Well, I'll tell you what the 3' end means, man. Look at this handy diagram on page 263 of your AP Bio textbook. You can see what I'm talking about, man. So here's the 3' end of the strand that's being created, and here's the 5' end strand. Now, what does that even mean? Well, that's talking about the order of the carbons in that 5-carbon sugar of deoxyribose. So the first carbon of that sugar is attached to the nucleotide. This is 1. This would be 2. This would be 3. This would be 4. And you see where this little L is? That would be 5. I'm sorry, Dr. Crisp, I got a little bit of pencil on your textbook. But we don't fuck with textbooks, boy! Anyway, so you have the 3' end, and then here's the 5' end. The 5' end is connected to the phosphate. This 3' end is where the new nucleotides, C for this example, would be attached. That phosphate would attach. Oh, wow, look at that. That is interesting. Now, why does it only attach to the 3' end? Well, that's the case for another video. Anyway, let's take a look at this freehand shit drawing that I made in 30 seconds. So you have the replication fork. Now what's this? Oh, this is helicase. What's helicase doing? Oh, it's unwinding the DNA. It's splitting it apart. It's jamming its finger up into the DNA, creating that fork, and making these two strands longer and longer by splitting up that DNA. Now, what is this? Well, this is a primer. Of course, you remember the primer has to start off DNA polymerase's job because DNA polymerase is lazy. So you have DNA polymerase with the primer, and it just starts working its way along this way. Now that's great because you can only add on to the 3' end of the strand. So it works out perfectly because it's going from the 5' to the 3' end. It can just keep on chugging along. And as helicase keeps on opening this gap wider and wider, it can just keep on going. It doesn't have to put in a new primer. That's why this is called the leading strand. Now I'll tell you why this one is called the lagging strand. So as helicase keeps splitting this DNA and these strands get longer and longer, of course the cell's going to put a primer, and then you got the DNA polymerase, and then it's going to keep on chugging along. Oh wait! What's this? You have a gap. Because as helicase makes this strand longer, and this DNA polymerase keeps on going that way, there's a gap. Now why is that? Because DNA polymerase can't work in both directions. DNA polymerase can only go from the 5' end to the 3' end. It could not add this way because it'd be going in the opposite direction. It can't do that shit. It can't, okay? It's a one-way street and you can't turn back. You can't go, all right? So DNA polymerase does its job. It's on its merry way, going from the 5' end to the 3' end. But then you got this gap right here. This gap where no nucleotides are being added. And what does the cell do? Well, it puts another primer. And then that primer can introduce a new DNA polymerase, and it can start chugging along and fixing that gap. But then another gap is created because the helicase keeps on going along and makes the strand even longer. So you end up with a bunch of these different primers that are being placed trying to keep up with the leading strand, because the leading strand only needs one primer. It can just keep on chugging along. But this lagging strand, it needs many different primers just to keep up with this guy. And these different sections, because it's got to have a bunch of different primers and polymerases working to complete this thing and add a bunch of new nucleotides, those different fragments, because they're going to be split up. They're going to look like this. This is a much better drawing. They're going to be fragmented like this. And these little fragments are called Okazaki fragments. The coolest word ever you'll learn in AP Biology. Okay, so you have these Okazaki fragments. Now, they're going to keep on going. These DNA polymerases are going to keep on going until they bump into the primer ahead of them. Now, what happens then? They're still fragmented. They're not connected. Well, that's when DNA ligase will come in and fix those gaps. DNA ligase is going to go along on the lagging strand and fix those little gaps once those DNA polymerases run into the primers. Back at the start of the video, you might have heard me reference DNA polymerase 1, and now's the perfect time to talk about it. So you have these strands, or particularly on the lagging strand, you have all these Okazaki fragments that are fixed by ligase. But then you have a shit ton of primers that aren't even DNA, they're RNA. Remember? RNA, I couldn't tell you why they are RNA, I don't fucking know, but they're RNA. So, you gotta get rid of them, and how are you gonna get rid of them? That's when DNA polymerase 1 comes into play. It'll go over those primers and replace that RNA with DNA. So, you're doing all this replication, you're creating these two separate helices, but then those helices are going to be intertwined. They're just going to be so twisted up and mangled and fucked up. Now how do you untwist those? It's basically like a circle and another circle, but they're twisted around each other, so it's impossible to get them untangled. Now, how are you going to do that? You're going to cut one of the circles, and then you're going to pull it through, and then you're going to attach it again. And who does that? Topoisomerase does that. It's going to fix all those tangled messes. So, DNA polymerase goes along, chugging along the DNA strands and adding in those nucleotides. Great. Go pat yourself on the back, bro. Go get yourself a beer. Go smoke a blunt. No, it can't. Wanna know why? Because it's not perfect. Nobody's perfect, okay? Let's not bash DNA polymerase. It's not perfect. It fucks up sometimes. And by sometimes, I mean like 1 in 1,000. It's pretty bad. But, you know what DNA polymerase does? It proofreads. It proofreads its own work. This tiny little enzyme proofreads. I don't even proofread my own essays, dude. And this little enzyme proofreads its own work. And that's great. But it doesn't necessarily eliminate the chance of errors. Because some wrong pairings can still go unnoticed by DNA polymerase. It's a little tipsy sometimes, okay? And what happens when that happens? Another enzyme, lo and behold, another enzyme or team of enzymes will go along, find that error, and fix it. Well, they won't fix it, they'll get rid of it, and then DNA polymerase will come along and fix it. Because DNA polymerase adds the shit. So they're called nucleases, and they'll go along and they'll snip off the fucked up part, and then DNA polymerase will come around round 2 because you fucked up the first time and do it right. So that decreases the chance of error to about 1 in 10 billion, which is close to nothing. So you don't have anything to worry about. Stop panicking. It's okay. So that was DNA replication. And hopefully I'll see you sometime next week to talk about gene expression.